those of you who don't know me, my name is Israel Lieberson. I'm the mentor for Elizabeth, who will be defending today her dissertation in the Neuroscience graduate program. Just for the order of things, Ellie will present. It's a public lecture right now for about 45 to 50 minutes. After that, there will be a short period of questions and answers and for everybody. After that, Ellie will step out for about 2-3 minutes. The committee members, which is Jim Abelson, Audrey Seasholz, Jack Parent here, Jeff Murphy, who is not present, will convey and will chat for a few minutes while Ellie is waiting, and then we'll invite her in and continue with the closed session of the defense, which probably will take at least another half an hour or so, maybe more, depends on the question and answer period. And after that, we will probably take another brief break and convene if we can with whatever our findings are. So people can schedule their timing. Now, we will not be able, because of the timing and holidays and all of that, we'll not be able to celebrate today, but we will celebrate it in January. Ellie is staying around, and if, regardless, we will celebrate the fact that she's defending. And, whether there's other things to celebrate, we'll see after that. That will be later on known. I'll let people know when the time comes toward definitely after holidays, probably second or third week of January will be my place. So without further ado, Elizabeth Rodriguez joined 6 years ago Neuroscience graduate program. She graduated from Hunter College in New York. She did a couple of rotations at the laboratory, settled out with us, and have been a member of our laboratory since then. And today she's going to tell us about the role of adult hippocampal neurogenesis and synaptic changes in mediating extinction retention deficits in rodent model of PTSD.
Thank you for that introduction. So I'll start with some business that is not science related, but is important to disclose, which is that our funding source comes from a DoD grant. Some of my education was funded through the Rackham Merit Fellowship, and I have no conflict of interest to report. And so why we're here is because I studied post-traumatic stress disorder in an animal model. And post-traumatic stress disorder is a debilitating psychiatric disorder that is characterized by the exposure, either witness or experience of an extreme traumatic event where the person felt extreme fear and hopelessness. This is followed by intrusive recollection in the form of flashback and nightmares. And then the person goes through great lengths to avoid any reminders of the event. It's also seen as hyperarousal symptoms. They're very alert, and they can be very easily startled. And these symptoms must last a month in order to meet PTSD criteria. This disorder is highly prevalent here in the United States, and it represents a public health burden. We do have interventions to treat people with post-traumatic stress disorder. The primary line of intervention is psychological intervention. And then the secondary line, if that doesn't work, is pharmacological, primarily antidepressants or SSRI. However, there's still a large number of people who do not see improvement in symptoms with these interventions. And those that do see improvement can still relapse later on, necessitating that we find modifiable targets in terms of mechanistic targets in order to make better therapeutic interventions for them. And so in the search for understanding the brain circuitry that might be affected in post-traumatic stress disorder, and that comes from a large work of human and animal research. And there's an overlap of the circuitry that is used or is important for contextual processing and that in which is seen in post-traumatic stress disorder, leading to the theory that perhaps a lot of the symptoms that we see in PTSD has to do with contextual processing deficits. And so what exactly does it mean to have deficient contextual processing? Imagine that you are in the wild and you come across a lion. Of course, it would be the appropriate response to be afraid in that moment. However, if you go to a zoo, even when they go through great lengths of pretending that you are back in the zoo, in the wild, you still understand the contextual cue led you to believe that you are indeed safe. And so a deficient contextual processing in this scenario would be acting afraid at the zoo. And so we have studied tremendously the safety and fear and safety learning, which is a very powerful tool. Specifically, it consists of placing the animal in a context made of different cues, smell, sight, etc. And here, if you can see they're placed in context A, where the tone is presented and paired with the shock, and the animal shows increased level of freezing as they learn that this cue is it's anticipating something adversive. Then the very next day, they're placed in context B, which is it's different from context A. And the same cue is introduced in this environment, but this time not reinforced with a shock. And the animal now encodes a completely separate memory of what the tone is supposed to signify within this context. And as you can see, their behavior, the freezing level goes down in this scenario, suggesting that they're encoding that memory. And so the following day, on the third day, whether you want to test them for extinction recall or fear renewal, you put them back into their appropriate context. And so if you place them back in context B, their appropriate response would be to not freeze, and this is contrasted by the fact that if they don't go through extinction, you see elevated freezing levels there. And if you place them, even if they have extinguished and encoded that memory, if you place them back into the original context in which they learned that it was dangerous, they will freeze again. And so we use that powerful tool, and we have developed an animal model of post-traumatic stress disorder in our lab that consists of the following 3 stressors. 2 hours of restraints, followed immediately by 20 minutes of forced swim. We allow them to recuperate, and then we put them in a chamber full of ether until they lose consciousness, and this can take about 5 minutes on average. And then after that, we leave them back into their home cage, individually housed for 7 days. And these are supposed to represent psychological, physical, and neurological stressors, all of which have been shown to activate maximally the HPA axis. And this was designed specifically to replicate post-traumatic stress disorder characteristic HPA axis dysregulation, with the hopes that we can then investigate further both behavior and neurological underpinnings of what we see with pertinence to PTSD. Since the development of this animal model 20 years ago, many labs have actually used our model and have contributed to the validity of the model. And so, and not surprisingly then, since I'm talking about contextual processing and fear and safety learning, what we found in our animal model is that they have no trouble encoding the fear memory. This is when they're trained in context A, the dangerous context. They have no trouble encoding the extinction memory, so pairing the presenting the tone repeatedly in a safe context. However, when we place them in either in a space in which they're supposed to recall either the extinction memory or the fear memory, they are unable to regulate their fear response and they show exaggerated freezing. And as I told you, these 2 processes are context dependent. The retrieval of those memories is context dependent. And so I go back to the neurocircuitry only to highlight the fact that I'm going to be talking primarily about the hippocampus, given its central role in this ambiguating and contextual processing, basically. And so one of the key features of the hippocampus is the sustained neurogenesis in adulthood. This is one of two well-established places or well-known places that it happens. And so all I want you to take away from this slide is that there are progenitor cells at the granular cell layer within the dentate gyrus of the hippocampus that express GFAP, and these are progenitor cells. They undergo differentiation and go into, I'm only going to highlight the neuronal cell fate, in which they start expressing double cortin as they progress. And then when they mature, they express mu-n. And their ability to encode LTP also increases as they mature and as double cortin expression goes down. And so the set of experiments that I devised were investigating the following question. Does adult hippocampal neurogenesis play a role in mediating extinction retention deficits? And for this, I did 3 sets of experiments. The first one addressing the following question. Does reducing adult hippocampus neurogenesis produce SPS-like extinction retention deficit? Secondly, does SPS affect adult hippocampus neurogenesis directly? And if so, does an environmental enrichment rescue SPS-induced neurogenesis reduction and extinction retention deficits? So for the following sets of experiments, this is the shared methods that I use. In the top panel, I'm showing double cortin labeling of immature neurons. This is a zoom-in version of, or zoom-in picture of the dentate gyrus, the hippocampus, showing the labeling. And I counted at 100x each of these beautiful neurons. And then at the bottom panel, I'm showing BRDU positive cells. And BRDU is a titamine analog that gets incorporated into the DNA of replicating cells, allowing us to mark when they're born. I am also showing you here that I only counted those BRDU positive cells that were immediately under the granular cell layer, and nothing that was obviously far from that. And so a reminder, the first question that I asked was, does reducing adult hippocampus neurogenesis produces SPS-like extinction retention deficit? And for this, I exposed the animals to 3 sessions of radiation for over 3 days. And then I allowed them to recuperate for 4 weeks before I trained them for fear and safety encoding and recall. And so I wanted to first make sure that the radiation did indeed work in reducing the number of immature neurons. And so I sacrificed the animal and stained for double cortin specifically. And as you can see, the radiation did work. About 70% reduction in immature neurons was seen on average. And importantly, we saw an extinction retention deficit resulting from exposure to radiation, or the reduction of neurogenesis within the hippocampus. Interestingly, the extent to which, so the number of double cortin immature neurons was negatively correlated to the amount of freezing during extinction retention. In other words, the fewer neurons they had, the higher the deficit they show. And so then this was promising, and so I wanted to look at neurogenesis effect, SPS effect on neurogenesis directly. And so I went ahead with the next set of experiments. For this one, I just exposed the animals to SPS, sacrificed the animal after the quiescent period, and assessed for double cortin expression at first. Specifically, RNA levels of double coordinating within the hippocampus. And I show here that SBS exposure did lead to a decrease in RNA levels of double coordinating. But I wanted to then make sure that this was a direct assessment of the number of neurons and not just RNA expression level changes. And so I repeated the same experiment, this time labeling, doing immunohistochemistry for double coordinating. And what I found was that SBS led to a decrease in the number of immature neurons in the hippocampus as a whole, but also when I further divided into dorsal and ventral, dorsal playing a role more in encoding sort of your surroundings and in positioning, and then the ventral has a greater role in anxiety-related regulation. And we see that in the hippocampus and in those two subregions, it's decreased, the number of immature neurons. And so I wanted to then have a more specific timeline of where SBS could be playing a role in or affecting neurogenesis. And so I looked at whether SBS could affect at the stage of proliferation. And what I did for this, I trained the animal, I exposed the animal to SBS again. I then, immediately after the quiescent period, injected them with BRDU. And right after the last injection, I sacrificed the animal and assessed for BRDU positive cells within the dentate gyrus. And what I found was that SBS exposure did lead to a decrease in the numbers of BRDU positive cells in this brain region, in the hippocampus. And that this change was primarily seen or contributed by the ventral hippocampus. But if you can notice on the top the timeline of when the neurons are going to be actually active and contributing, and you think back at the deficit that we see following post-traumatic SBS, it's a timeline of only 11 days. And so I wanted to then ascertain, well, could SBS have an effect on the survival of neurons or later stages in the development of these neurons? Oops. So pretend you didn't see that. So what I did here was I treated the animals with BRDU again, and this time I waited about 4 weeks to expose them to SBS, and then sacrificed the animal after that to measure the number of BRDU positive cells. And since you see this for the first time, I'm sure you're shocked that SBS did lead to a decrease in the number of BRDU positive cells. And that, again, this was primarily contributed by the ventral hippocampus. And so the last experiment that I did was to truly understand the relationship of neurogenesis in this deficit. I wanted to see if does environmental enrichment could rescue, and this is an intervention shown to promote survival of this newborn hippocampal neurons. Does environmental enrichment rescue SBS-induced neurogenesis reduction, thereby rescuing perhaps the extinction retention deficit we see? And so for this, very similar to the survival experiment that I did, I injected them with BRDU, then exposed them 4 weeks later to SBS, and after the quiescent period, they got to play with toys in their cages. I gave them 3 toys at a time, switched them every 2 days to keep it fresh and new. And this is more or less what they were playing with. And so what we see here, the plain ones are without the environmental enrichment, and the checker ones are with the environmental enrichment. And as you can see, SBS exposure led to a significant decrease in the BRDU positive cells. And that environmental enrichment brought it back to within normal range or control, close to controls. And this was seen in the sub-regions as well. Importantly, we see here that SBS did lead to an extinction retention deficit. and that environmental enrichment also was accompanied by alleviation of this, rescuing of this deficit. So brought it again down to normal. And importantly, just as I showed you with the radiation experiment, was that the number of BRDU positive cells was negatively correlated to the amount of freezing that they exhibit during this time. Or in other words, the fewer neurons they had, or the fewer BRDU positive cells, the greater the deficit during extinction retention. And so this sets of experiment showed you that impaired adult hippocampus neurogenesis may underlie extinction retention deficit in our single prolonged stress animals. Because reducing adult hippocampus neurogenesis produces extinction retention deficit, and SBS exposure affects adult hippocampus neurogenesis during both proliferation and survival. And I also show you that an intervention aimed at promoting survival of adult hippocampus neurogenesis can improve SBS extinction retention deficits. Of course, as strong as I like to think that these experiments are, the body of them, there's still some caveats to each of them. The first one is the fact that the radiation is not a non-specific treatment. And so I would love to repeat that experiment using a GFAB transgenic rat, I'm hungry, guys, rat, where I target those progenitor cells specifically and see whether the deficits can be replicated. I also understand that BRDU is non-specific, and that I would have to also do double labeling with a neuronal marker to make sure that the populations of neurons are being affected, and to what extent. And lastly, environmental enrichment could rescue these effects through a separate mechanism. And so I would also utilize the GFAB transgenic rat as a control to eliminate neurogenesis and see whether rescuing still remains. But the future direction, let's say that neurogenesis is a player in mediating these effects. The future direction would be to look at the mechanism by which SPS affects neurogenesis. And so these are just 4 that I could come up with, but I'm sure there are more. One of them could be stress hormones mediated through GR, programmed cell death through different pathways, it's cytotoxicity, and GABAergic mediated. And so the second set of experiments that I'm going to be talking to you about is also looking at the mechanism that could potentially mediate these deficits. But now I'm going to focus on synaptic changes within fear processing brain regions. And for this, it's exploratory, keep in mind. I only assess mRNA expression levels within the medial prefrontal cortex, the hippocampus, and the amygdala. And for this, I used real-time quantitative PCR. I ran them in duplicates, and I did it twice. So I was able to obtain 4 values. And I had at least 3 values that were within 90% of each other. I normalized them against the housekeeping gene, HPRT. And all the results I'm going to show you are going to be represented by 100% controls, and then SPS a variation from that. Here I just want to orient you to the different categories of synaptic genes that I assess. You don't have to remember any of this except for what I'm about to tell you, which is the first one is the growth promoter, BDNF. Then there were genes related to dendritic formation. Then there are genes related to vesicular trafficking and docking. Then the next one is MAGUX, or scaffolding postsynaptic density proteins, as well as major receptor subunits, AMPA receptor, and MDA receptor, and GABA-A receptors subunits. And I'm giving you everything in this slide. The first set of graph is the medial prefrontal cortex, in which I found that an mRNA expression of BDNF went down. The morphological or branching gene MAP2 kind of went down. And the COM2, which is related to vesicular function, as I mentioned, went up. Similarly, in the hippocampus, MAP2 went down, and GAP43, which are all dendritic formation related genes. And the two vesicular genes that we found here went up. In the amygdala, we saw the most changes, where we saw two dendritic formation genes. One went down and another one went up. COM2, as in the other two regions, went up. And this MAGOOC went up. And interestingly, AMPA receptor subunit went down, while the GABA A receptor subunit went up. And just to summarize, this is what I have come to think about these results. It seems that SPS decrease structural markers while increasing functional markers, perhaps in order to compensate for this loss. And that in the amygdala, there seems to be a shift from excitatory to more inhibitory neurotransmission, or signaling specific. Of course, there are many caveats, given that this is PCR and I'm just looking at RNA levels, mRNA levels, is that these are not that proteins were not directly assessed, and that I am combining subregions within these regions that I assess that have opposite effects on the regulation of fear. Specifically within the medial prefrontal cortex, the prelimbic promotes the expression of fear, while the infralimbic sort of dampens it dampens the expression of fear. In the amygdala, similarly, the BLA promotes the expression of fear, while the intercalated cells through the BLA are supposed to regulate the expression of fear. And that these are just a few of the many genes that could potentially play a role in synapse and synaptic function, and many of them remain unknown. So, of course, I would love to assess more directly both the dendritic morphology that could be affected by exposure to SPS, as well as the relative numbers of the different synaptic types within these regions. But more important than that, I want to know how the brain regions are talking to each other. So I would love to inject a tracer in a brain region, see where it goes, and, well, pertinent subregions where it goes, and see not only the connection, but also the architecture within those connections, and assess the number of synapses there, as well as their function. And you can do that with immunohistochemistry and other techniques that are now advancing. So with that, I would like to first thank my committee chair and my mentor, Israel Lieberson, for not giving up on me, primarily. Secondly, for encouraging me, and for always remaining excited about research, and I hope I remain that way as well. I would also like to thank Jim Abelson, who's sitting back there, for all his help, especially in the last year, helping me learn how to write, and I know it wasn't an easy task, but I thank you for that. And to the remainder of my committee members, I'm sorry I didn't take as much advantage of the wealth of knowledge that you have, and I regret that. But I'm thankful for the times that I did tap into those resources. I would also like to thank the rest of the Lieberson Lab team, both past and present. John Greco helped me pretty much with every single experiment that I show you up there. Nirmala was just there for anything and everything that I ever needed. And then the rest of the members, I'm sorry, I'm not going to go through each one of you, just know that I'm thankful. I would not be here if I didn't have an awesome relationship with the staff members at the BMU, Sherry, Maurice, Hal, and Rosemary. And of course, to the Neuroscience program, Ed Stunkel, who is the director, Audrey, who is also works there, and Valerie and Rachel, who, I mean, I don't know how I didn't drive you ladies crazy, but I truly appreciate all the help. Of course, my journey didn't start with graduate school. It goes way back. This has been my long-term dream leading up to this moment. So I started at Hunter College through Mark and BRS programs. Dr. Lipke, the director, sat with me and we studied organic chemistry together. My first mentor, Dr. Hardin, who introduced me to research for the first time, we studied sexually dimorphic behaviors in birds. There she is, and there I am. And the summer programs, where I was also introduced to different kinds of research. Then I wasn't feeling up to it, so I did a post-bac program at Columbia University, where I worked with Dr. Silver, where I study circadian rhythms and the new hot topic, which is how immune system and nervous system talk to each other. So that was really exciting, and she encouraged me to be the best scientist I could be, as well as other mentors here within the university. My friends keep me sane. They fail at that sometimes, but I thank them anyways. My best friend Denise, who is not here, she's probably the silliest person you'll ever meet, but also the most dedicated scientist, young scientist that I've ever met as well. My friend Ramon, who came from Rhode Island to be here, thank you. You've been a great emotional support for me. And of course, my family. This is a picture of us back in the Dominican Republic. And my two sisters. And I'll take a second, if I may, just to illustrate how important and where my inspiration has come from. My younger sister was born with a cognitive deficiency, and that later became a schizotypal disorder when she was a teenager. And then that became schizophrenia and bipolar disorder. So I remember one instance when I came from Hunter and I was already doing research, and our one-bedroom apartment was covered in nonsensical biblical writing everywhere. And I noticed that there was these little jars of garbage in my mind, in little jars of water.